Excitation of the muscle cell or fiber involves the creation of action potentials that spread along the length of the sarcolemma. So the end plate potential that was started by the acetylcholine that opened the acetylcholine receptors allowed sodium into the cell, and that end plate potential spread to adjacent regions of the plasma membrane, where in these adjacent regions it stimulates voltage-gated sodium and voltage-gated potassium channels. So these voltage-gated sodium and potassium channels are the channels that are responsible for creating action potentials along the length of the plasma membrane of our skeletal muscle cell. So if we focus on this small region of the membrane, the voltage-gated sodium channels are the ones that open first during the action potential, and these allow sodium to enter the cell and depolarize it. So that means that the membrane voltage inside of our skeletal muscle cell becomes positive in that area for a brief time. Then after a little bit of time has elapsed, and we're talking thousands of a second here, the voltage-gated sodium channels will close, and then voltage-gated potassium channels in that same region will open, and these allow potassium to leave the cell, and the cell repolarizes. So that means that the voltage inside of the cell or inside the membrane at that point is going to drop back down to its resting membrane voltage. So we need voltage-gated sodium and potassium channels in order to create the action potential that spreads along the skeletal muscle cell. So if we were to measure the membrane voltage in this small region of the sarcolemma during the action potential, this is what we would see. So in this graph, we have this kind of hill-shaped graph here where we see an initial uphill phase and then a downhill phase to end that action potential. We have membrane potential or voltage on the vertical axis and time on the horizontal axis. So notice that time is in milliseconds here, so this is thousands of a second that we're talking about. So one action potential in one small area of the membrane is over in roughly, seven or 8 milliseconds after it begins. So that's 7 or 8 thousandths of a second after it is started. So these are very rapid changes in membrane voltage. So what we're seeing here again is what we had discussed in the previous slide, is that the cell goes from a resting membrane voltage of negative 90 millivolts at rest to roughly a plus 30 millivolt peak during depolarization. So after the cell is stimulated, it will go from rest. The voltage-gated sodium channels will open. This allows sodium to enter the cell, and we see that the voltage climbs past zero, from negative past zero up to plus 30 very rapidly. So it only takes a couple of milliseconds for that depolarization to occur. So this is when sodium is entering through voltage-gated sodium channels, and we see the voltage increase from negative 90 to plus 30. On the other hand here, the downhill portion of the graph is when the cell is experiencing repolarization. So what we're seeing is that the cell is going from plus 30 millivolts back down to its resting membrane voltage or resting membrane potential at negative 90 millivolts here. And this is during repolarization. So what's happening at the end of this action potential here is that those voltage-gated sodium channels are closing, so sodium is no longer being allowed to enter the cell through those channels. And instead what we see is that potassium is allowed to exit the cell through voltage-gated potassium channels. So in the repolarization phase, potassium, which is a positive charge, is leaving our cell, and that's what is causing the drop in membrane voltage from plus 30 back down to the resting membrane voltage of minus 90 millivolts. So because voltage-gated sodium and voltage-gated potassium channels line the length of the sarcolemma that is beyond the neuromuscular junction, action potentials spread from the neuromuscular junction to essentially all of the, every inch of that, or every millimeter, if you will, of that plasma membrane, due to the presence of those voltage-gated sodium and voltage-gated potassium channels that are found along the entire length of the skeletal muscle cell plasma membrane. And we see that the action potential spreads like a wave across the plasma membrane of our skeletal muscle cell. And this spreading of the action potential is referred to as propagation. And so what we're seeing here is we're seeing kind of a time lapse of, so if this is the neuromuscular junction over to the left, I kept it out of the other part of this diagram just for simplicity here. But what we're seeing is a length of the plasma membrane that's extending beyond the neuromuscular junction here. And we see that in blue here, the depolarization phase of the action potential is occurring. And in green is where we see the repolarization phase of the action potential. So in the depolarization phase, voltage-gated sodium channels are open. That allows sodium into our cell, and we see our voltage inside of the cell at that particular point rises to a peak of about plus 30 millivolts during that action potential. What you're seeing in this diagram here, since it's a time-lapse image or diagram of this same region of the membrane, is that this region here, which a little while ago or a few milliseconds ago was just undergoing a depolarization, this region is now undergoing a repolarization where voltage-gated sodium channels are closed, so sodium is no longer entering the cell at this region, but we see that voltage-gated potassium channels are open and potassium is now leaving the cell, and that's causing our membrane voltage to return to minus 90 millivolts, which is our resting membrane voltage. The segments that are in red here on either side of where the action potential is occurring are indicating resting membrane voltage or potential here. And we see that the plasma membrane spreads like a wave, right? So the action potential that begins here, the depolarization that starts here, triggers a depolarization in the adjacent region over here. And so we see an action potential starting in this region to the right of this action potential here. And then a little while later, we see an action potential occurring in this region because it was stimulated by this action potential that was starting there. So we see that the action potential spreads or propagates like a wave across the entire length of the plasma membrane thanks to the presence of those voltage-gated sodium and voltage-gated potassium channels.